Hey, hello. Once again, I am Danica. Welcome to my TED Talk this week. In the interest of bad science, I thought I would cover the topic of how women have essentially been excluded from a lot of studies, and we're going to go in depth about how that has been an issue from bench to bedside. So it's kind of the correlation between excluding sex as a biological variable in basic science and how that has essentially affected the way that we practice medicine and how that has consequences for that translational aspect of patient outcomes. So SGSH, or sex and gender specific health, seeks to understand differences in diseases between men and women. And the goal of this kind of field of research is essentially to improve patient outcomes by applying a sex and gender specific knowledge and approach to this type of biomedical practice. So these sex-based differences exist in disease epidemiology, presentation, pathology, prognosis, as well as response to interventions, and obviously the resulting outcomes. Therefore, the way that prevention and treatment are approached for men and women should thus cater to their respective health needs based on physiology and the other interactions with their environment that are affected by sex and gender. So it's going to be important to note that in addition to sex itself, the age group and the time point in which we see the onset of certain diseases and symptoms and whatnot will vary between the two. And this is essentially attributable to a variety of different factors, but one that's often overlooked is the hormonal profile of women and the estrous cycle. And it's going to be kind of interesting as we go in today and delve into the history of how women have been excluded or ostracized in biomedical research based on, their reproductive system. And we're talking about like the Hysteria Discourse, how that has kind of laid the foundation for the oppression of women in society and especially in biomedicine. So we're going to be covering that today. Oopsies. All right, so as always, we're going to start off with our presentation outline objectives. So we're going to start off by defining sex and gender, as they are obviously two different things. We're going to go into an overview of the exclusion of women and females in biomedicine. We're also going to then talk about the whole process of exclusion and process of sex bias from bench to bedside. And we're going to talk about how some of these biases have tried to be improved or increase the awareness and consideration for this, thanks to the NIH policy for sex as a biological variable, or SABV. A quick overview of why it is important to include this in research, and we're going to go looking at specific examples in cardiovascular disease. So I have like 3 specific examples we're going to go through. A quick wrap up with sex differences in reporting for patient outcomes, and then future steps of how we can further tackle this issue moving forward. So, without further ado, we will start off with defining the difference between sex and gender. So sex contributes to biological and physiological differences, which influence disease onset, risk factors, severity, comorbidities, signs and symptoms. So at TIM, this is like a really great opportunity to kind of think about the considerations of this with respect to pharmacokinetics and pharmacodynamics. How drugs are metabolized, absorbed, distributed, these are all going to be an important component of the definition of sex in biomedical practice. And hormonal effects across life stages are also going to be important for mediating these sex differences. So understanding the entirety of the individual, considering both sex as well as gender, is going to be really important for how we can take our research findings and apply them to the clinical setting and improve healthcare. So obviously, we can see here, sex is defined as the biological variable assigned by chromosomal complement. So X chromosomes and Y chromosomes, obviously XX for female, XY for male. And then these differences in terms of sex are defined in context of their sex organs, physiological functions, as well as the hormonal profiles that are associated with either like male or female. On the other hand, gender is a social construct. It's defined by roles, behaviors, and expression, and identity. So these are essentially things that are placed on the individual based on what society determines to be appropriate for that category or that box. And although it is not, I guess people could, I guess, traditionally say that's not like an actual, like a physiological component, as we're now starting to understand the significance of physiology and health relative to environmental interactions. These are obviously going to be things and factors that will affect one's health based on the interactions with their environment. So the big takeaway here is that understanding sex and gender-based differences will help us better address the differences in disease, incidence, severity, and response to treatment. And ultimately, we want to be able to round this out in a way that will improve and ensure equitable health for all individuals, regardless of their sex or their gender. There's especially a dire need for new diagnostic tools that work for women. We're going to go a little bit more in depth about this later on to go through the examples with our cardiovascular disease specifics. But that's just kind of the main takeaway, that the exclusion of women and overlooking these, I guess, concepts of sex and gender have resulted in a lot of these differences being overlooked and poorer health outcomes for the excluded population, or in this case, women. Okay, so as with many things that are in this world, the system was built by men for men. So we're now going to delve into a brief overview of the history of the institution of patriarchy in biomedicine and women's health, courtesy of The Guardian and a few other sources. So first off, we're going to enter Aristotle, who actually characterized the female as a mutilated male. And these views were implemented into biological, obstetrical, and theological traits. And his theories on reproduction centered around the male actually contributing more or greater to the process of reproduction. And therefore, he kind of defined women relative to man as lesser, as weaker. And, you know, it's interesting because now we obviously know the progress of biomedicine, that it's kind of the opposite, in which the oocyte and the egg and the embryo and everything is housed in the woman and grown and whatnot. But this misinterpretation of that back in the olden days has kind of contributed to this very patriarchal woman less than man ideology that has influenced and progressed and persists in Western biomedical practice. So now we're going to kind of move into discussion about the historical hysteria discourse. So women were often and pretty much always endorsed as difficult to treat because of, you know, the fact that they were women. And instead of actually addressing the limitations on medical knowledge at the time, responsibility was in many ways put on women to accept their illness, to make their own lifestyle changes. And if we're even talking about this just in the meta kind of like scheme of how the idea of like changing individual behaviors is going to somehow magically solve these problems, no. Like if we look at it in the context of like biomedicine and the public health perspective, typically, yes, it's going to be a factor and a component of how changes in individuals' lifestyles will improve outcomes, but there typically is an indicator or proxy for there being some underlying upstream effects that are causing these issues. So that was kind of one preliminary example of how this was an issue with how women were interpreted and how they were then, the burden was then put on them as individuals for their own health and disease. So the female body itself has been defined throughout history as distinct but as not equal. And thus the problem is that if we are setting male physiology as the standard, as the template, against which we judge health and disease, any aspect of the female body that differs from the male or that cannot give a male comparative is always going to be viewed as evidence for deviation. And in the following slide, we'll delve a little bit deeper into this and hysteria as a discourse, but essentially hysteria was used as a catch-all term to categorize any of these deviances from these male set benchmarks. And so when symptoms were presented that were unique to women that we couldn't find a comparative to men, it was always just kind of overlooked and seen as something that we couldn't and should not address. There has been some progress made or some attempts. So in the 1980s, a group of female scientists banded together to form the Society for Women's Health Research. And they worked alongside the US Congress to draw attention to discrepancies in medical research and the effect of women's health. In 1985, the US Public Health Service Task Force on Women's Health published a report that warned of historical lack of research on women's health, resulting in the quality of health information for women and the healthcare that they received as being compromised as a result of that. And for example, it was observed that women tended to have lower rates of heart disease until their estrogen levels dropped post-menopause, but because they weren't studied long-term, this wasn't a fact that we had considered to be important. And there have been a slew of different studies that have just outrightly excluded women and have really defined bad science with the exclusion of women and sex as a biological variable. So there was a 1986 study by the Rockefeller University which wanted to look at the correlation between obesity and breast and uterine cancer. And guess what? They actually did not enroll a single woman. So it was really ironic that they did not enroll a single person with a uterus when they were trying to study uterine cancer. And it was actually quoted by one of the scientists working on this project, Mr. Leon Bradlow. He commented that the reason why they would prefer to study men was because they were cheaper, because they were hormonally simple. So take with that what you will. Other studies included the Baltimore longitudinal study on aging, which began in 1958 and did not enroll a single woman for the first 20 years of the study, which is, again, just been a testament to the lack of our information and understanding of how we've characterized women's physiology. And there have been other studies that have essentially found that, in providing aspirate every day is supposed to prevent cardiovascular disease in men, didn't test women. So again, that's just kind of the standard that is set, but we'll later find out that there are fundamental differences between both men and women at almost any organ level. And then finally, one last example I'll make is between 1997 and 2000, 10 prescription drugs were actually removed from the market by the FDA, and 8 of those 10 were actually found to have severe health risks, like significantly more severe health risks for women as opposed to men. But again, this just kind of goes to show that because studies have been skewed at every level, especially at the clinical trial level where women have not been included, we are missing the fact that they could be put in a greater health compromised situation because they have been excluded. Okay, so we're just going to take a quick little pause to talk about the hysteria discourse for a brief moment, as this kind of sets the foundation, the precedent for what we can understand of why women have been excluded and how this has affected almost every aspect of society, especially biomedicine. So back in the olden days, any symptoms, so fatigue, seizures, paralysis, pain, even rebelliousness, was categorized under this catch-all term of hysteria for diagnosing women. And this was a term essentially coined and constantly reiterated by both physicians as well as philosophers. Shout out to our boy Aristotle. And hysteria itself actually comes from the Greek word for uterus. So as if we weren't already putting enough blame on women. And today in clinical practice, women actually still suffer from this trust gap in which this long history of omission and not believing that women's actual experiences and symptoms are legitimate. Anytime that a woman can try to convey or communicate the symptoms that she experiences with any disease and illness, there's still a huge bias in our world today that will discredit a woman or say that the authority over her understanding of her health is less than what a man knows about his own. And as a result, our lack of proper diagnostics and treatments for women and the consideration or lack of consideration of women's health can be attributed to these dark origins in which hysteria would be treated with leeches on the vulva and injecting the uterus with all these invasive and strange chemicals. And this perception is also kind of spoken to these differences that we've seen not only in sex, but also in class and in race. As if we look back to those, ancient, not so ancient times, hysteria was actually used as a way to diagnose specifically upper class, native-born white women, where they were prevented from doing work because their bodies were incapable. But if you were someone who was a non-white woman, you'd be less likely to be diagnosed with hysteria because that would essentially allow you to continue to be overworked and to be used for experimental procedures. So there's also not only a long history of sexism, but also classism and racism, as there is with everything in this world. So this enforced patriarchal institutions and men to keep women out of medical education. So outside of just the studies, women were also locked out of the system itself. And obviously without women to kind of speak to, represent, and study issues and things that were relative to their own bodies, that aspect of science and biomedicine and physiology was never properly understood. And that's why we have this huge lag and delay in our understanding of our own physiology and anatomy. So keeping women out of science also resulted in a lot of women kind of feeling the sense of deficiency on being unable to contribute equitably to society. And as a result, it has not only affected our participation, engagement, and contribution to biomedicine research, but even the political sphere and our ability to contribute to society as a whole. So on every level, we've been oppressed. So now that I've kind of gone over the history and had my rant of oppression of women, at every level of society, we're going to talk a little bit more about how this directly applies to science and its biases and our understanding of biomedicine and healthcare. So we actually see bias at the level, at essentially every level from bench to bedside, in every facet of research. So according to a study from 2015 under the United Nations Department of Economic and Social Affairs, women in developed countries make up approximately 51% of the population, and 80% of healthcare decisions are made by women. That's both in the context of, their largely proportional contributions to healthcare as a whole, but also women are also typically the primary decision maker or stakeholder in decisions around health regarding their families. And despite this fact, and despite the representation of women in society as well as in healthcare itself, translational research has omitted them vastly. So 76% of studies involving cells or cell lines did not report the sex of the cell. And there are several lines that are only manufactured and derived from male cells. In animal models, which provide essentially the foundation of the majority of biomedical research, 80% of the studies only used male mice. And clinical studies are also known to be biased and skewed, especially in clinical trials of cardiovascular disease. 67% of the subjects studied were men. So if even by percentage and numbers we are omitting women and what their physiology and contributions to this research could basically contribute to the advancement of science, not having or just completely overlooking or excluding this factor as sex as a biological variable means that we're missing this entire realm of understanding of it. So what do we do about it? Enter the NIH, thank goodness. Shout out Risa. In 2014, and Soren, because y'all both did NIH things. But in 2014, the NIH began to acknowledge this problem in male bias in preclinical trials. And then a year later in 2015, they actually released a notice outlining the expectation that sex as a biological variable, or SABV, would be factored into the research design, analysis, and reporting in vertebrate, animal, and human studies. And then, hallelujah, in 2016, it mandated that all grants that will be receiving funding, essentially, they will only be provided if female animals are included in the study / included sex as a biological variable. So they were really trying to push this policy and kind of backed it up with the sense of funding to ensure that we were getting more holistic understanding of biomedicine and physiology by including sex as a biological variable. So what is the significance, or why is this important? Why should we care? Several reports have essentially found recurring themes in determining the importance of sex differences and SABV in research. One, differences in health and illness are influenced by individual genetic and physiological constitutions and interactions with the environment. So incidence as well as severity of disease will vary between sexes, and the time point in which the onset of these diseases and symptoms occur is also in accordance with the factor sex as a biological variable. Two, the study of sex differences is evolving into a mature science. We are moving away from just descriptive to experimental, from characterizing to actually trying to understand the molecular basis of these differences, and we're trying to determine the mechanisms and origins of sex differences. So I'm just going to reference really briefly kind of like a personal connection to what this means with respect to like my research. So I know that in my lab, as we're looking at cardiac lymphatics, we are very strongly looking at the differences between male and female phenotype of the vessels and the branching patterns and whatnot. And initially we'll start by looking at just characterizing the phenotype, what is the area of coverage of these vessels, how wide are they, et cetera, some of the more descriptive qualitative aspects. But now we are trying to kind of move away from simply just characterizing it into actually trying to define, are there differences in expression levels of certain genes between sexes and how that might have an impact. So that is also something that is more widely being applied in research today. And then three, barriers to advancement of knowledge about sex differences in health and illness do in fact still exist and must be eliminated. And the first step to solving our problem is admitting that we have one. So just to kind of speak to a little bit more of the specifics of this, genes on the sex chromosomes can and are expressed differentially between men and women. There are several X-linked genes that will result in differences due to double or single copies of certain genes in their expression. It's good to kind of determine whether the differences that we see between the two are X-linked and attributable to the chromosome themselves, or if there's some other factors such as the hormonal profile. And it's also with respect to the hormonal profile to think of that into consideration of at what time point will we see these differences developing. Will we look at the embryonic stage as a baseline? Will we see how that puberty stage will be affected with the induction of these higher expression of the hormones? And especially when we look at women and the studies of postmenopausal, is there a cardioprotective effect or another physiological effect that has been protected because of this hormonal profile that is later then diminished postmenopausal in women. So that's also an important consideration to have. And males and females fundamentally have different patterns of illness, different lifespans, and better understanding the reason for this and the interaction between various genetic as well as environmental factors from the gender side of it. This will actually allow us to better approach prevention, diagnosis, and treatment in research as well as clinical practice. So a few examples here, sex differences have actually been identified in brain tissue. So there's been differences in neurohormonal signaling. This has also actually been tied to a lot of the cardiovascular regulation. Kidney cells, for example, when they've conducted research looking at some of these cells, they've actually found differences not just in the cell itself, between sexes, but also in the way that they're treated. So if you're to treat, I think, embryonic kidney cells with ethanol, female cells actually tend to be more sensitive to the response. So if you're trying to get the most accurate results and do a comparison, you want to make sure that your cells are viable. So you also have to be cognizant of how you run your experiments and how those might differ and those variables might differ between the sexes. And they've also found differences in protein expression levels between liver cells of males and females. That's also another important consideration to take account for. Another example we want to think about is the HeLa cells lines. So HeLa cells have been used for decades and decades now, derived from Henrietta Lacks. And this also kind of brings up another issue of, classism, sexism, racism in science itself, as these cells were taken without her consent. Anyways, we haven't fully understood or characterized the profile of these cells genetically, though we've been experimenting with them for decades, and they've basically been used to push forward the progress of a lot of biomedicine, but we haven't even really considered, what the implications might be of that sex profile of the cells. And then finally, the last point I'm going to make here is that sex differences are observed in cells from different tissues. So it's going to be important not just to think about the sex, but the specific tissue it comes from, and not only the differences in the tissue, but the expression levels of proteins, enzymes, et cetera, et cetera. So now we're going to talk a little bit about some specific applications in cardiovascular disease. And there are three kind of major examples I'm going to make in today's presentation. So just as a brief overview, cardiovascular disease, or CVD, kills more women in the United States than any other disease. And it's predicted that half of all American women will develop some form of heart disease at some point in their lifetime, whether it be ischemic or non-ischemic. And environmental factors like stress and poverty differ for women and can in fact cause differences in expression, diagnosis, treatment, and outcomes of CVD. Cardiometabolic disorders of pregnancy, so things that include preeclampsia, gestational diabetes, as well as hypertension, also associated with pregnancy, can increase a woman's risk for developing CVD and can increase the risk of CVD in their children. So in terms of just a broad overview of some differences we see between men and women and CVD, is that women tend to develop ischemic heart disease on average 10 years later than men. And it's been suggested that the hormonal profile with estrogen and whatnot, and then the drop off of that with postmenopause has kind of contributed to this variation or this disparity. But younger women who experience myocardial infarction or heart attacks actually have higher mortality rates. And it kind of goes to the question of, does this speak to the fact that we don't properly understand the physiology and the pathophysiology in women as well as we do in men, because they haven't been characterized or studied as much. And then it kind of begs the question of, how does our understanding of basic science and then how that translational piece affects clinical practice, if that itself is failing women in our healthcare systems. So we do know that postmenopausal women are less likely to develop CVD. And currently there's actually not enough research that exists on estrogen receptors in the smaller blood vessels, which is actually where a lot of the disease-producing factors are in women. And we have also found that hormone therapy including estrogen and progesterone in menopausal and postmenopausal women, can increase the risk of stroke and pulmonary embolisms. So kind of trying to understand, how well have we characterized or understood these standardized treatments, and are they even effective and safe for women is one of the major questions that we're faced with tackling right now. And again, time point considerations is kind of what we're talking about here on this graph. So if you can see in figure 1, it really shows that, figure 1 talks about the publications. So if we look at the amount of data and publications that have been conducted on coronary artery disease itself, you can see that there's a big disparity in the data that's been collected with and without women, which kind of shows, how this will affect your ability to properly practice science, research, and, medicine. And then as you can kind of see here in this, I guess, figure 2, I didn't label it, sorry. There is a difference in the actual process of heart remodeling with aging between men and women. So what we'll kind of see is this concentric remodeling in women, where we kind of see this normalization of increased wall stress in the female physiology, whereas we look at men, their physiology kind of allows for the walls to remain thin, for the actual volume to increase, so that we still have a sufficient amount of the total stroke volume in men. So that just kind of goes to show that there is a fundamental difference between the anatomy and physiology of each sex and how that can then affect the way we diagnose, treat, and prevent CVD. So our first example is going to be HFpEF, or heart failure with preserved ejection fraction. So in this disorder, there's an increase in stiffness in the left ventricle, which can cause stiffness and consequently decreases the left ventricular relaxation when the chamber fills. So it's actually a disorder of diastolic dysfunction, or disorder of diastole. And this results in increased pressure and or impaired filling of that specific chamber. And ultimately, a stiff, non-compliant heart can result in blood flowing back as if it were hitting a wall, and it can actually enter back into the lungs, which is why one of the symptoms that we see of HFpEF is pulmonary hypertension. So it is associated with pulmonary hypertension, dyspnea or trouble breathing, swollen ankles, abdominal bloating. So there is a component of edema here in various organ beds. And studies have actually identified that women have a higher prevalence of hypertension and atrial fibrillation in this disorder, while men have a higher prevalence of coronary and peripheral artery disease. So this study here, that is the graph featured by Oslik et al., essentially found that with respect to signs and symptoms, women tend to have more symptomatic, are more symptomatic for heart palpitations. They experience reduced exercise tolerance. They experience peripheral edema, fatigue, and with respect to treatment itself, they were less likely, so women were less likely to be administered beta blockers and antiplatelets as compared to men, where women were just given anticoagulants because there was a misdiagnosis of this specific disorder. They also concluded that women were ultimately more symptomatic, had higher BMIs, had worse diastolic function. This may be attributable to the difference I mentioned earlier with the concentric development or changes of the heart with aging. And these women actually tended to be older, whereas male patients tended to be younger. So that's why it's also kind of important to understand, we have to follow the timeline, we have to follow the life cycle and onset of the disease, because if we're setting the time point or a cutoff point in accordance with the male, for example, at this point where we kind of see it dip off, which is the point at which the diagnosis increases for the other sex, we're missing this entire population of patients if we're setting the benchmark where we just start to see it drop off for men. So that's an important consideration. So how can we address some of the key knowledge gaps? Well, one, with respect to HFpEF, we can look at preclinical study design. We have to make a requirement for better animal models. As we said, majority of the studies that are conducted only include male mice. And even just trying to include more female mice at the experimental design level, that itself will make a huge difference. clinical study design, there's a lack of sex-specific data on comorbidities and outcomes. So it'd be important for us to characterize even mild phenotypes that are associated with men versus women and how those can also be monitored and implemented into clinical practice. At the hormonal level, estrogens trending in a cardioprotective fashion, that's great for what we understand up to the point of, premenopause. But if we're not following that timeline postmenopausal, then how are we supposed to understand how to better address or treat those differences in the patient? And then again, in terms of the phenotype, if we're setting the benchmark at the male kind of exhibition of symptoms and physiology, we're not going to be able to account for those discrete phenotypes that may present in women as opposed to men. So those are going to be some important things to consider with respect to this disease as well as others. So the next example we're going to go over is CMD, or coronary microvascular dysfunction. And it is actually as common as HFpEF, but it is far less well characterized and understood. And it's actually pretty important for identifying patients with symptoms suggestive of CAD or coronary artery disease. So looking at this or looking for CMD is kind of what we look for in advance or before we get to the point of coronary artery disease. So CMD itself is a cardiac ischemic disease, which is defined by a lack of adequate blood supply needed to meet the blood supply demand by the heart. And in men, this tends to be present as a blockage of the large arteries outside of the heart, whereas in women, it tends to be a disorder or a disease of the microvessels, so the smaller vessels that actually penetrate into the heart. And the biggest issue here is that because our diagnostic tests and whatnot are kind of focused on looking at these obstructive indicators in men, it's very common for us or for clinicians and whatnot to miss this microvascular disease in women. And in many cases, it goes misdiagnosed or undiagnosed for a longer period of time, which can lead to complications and therefore worse health outcomes for women. So the main thing here is that angiograms are typically used when we look for CAD, but they are not sensitive enough to detect CMD. And therefore, patients with signs and symptoms of ischemia, but no obstructive coronary artery disease, which is the majority of women who present with this, actually are not getting their problem resolved. So if anything, at the moment right now, where we are in terms of science, CMD can only be tested through very invasive reactivity testing, which involves injection of substances like adenosine, acetylcholine, and nitroglycerin. And these can only be conducted in highly specialized and advanced centers. So this also kind of talks about, lack of accessibility to the ability to be diagnosed properly for women, which is, again, like another barrier and hurdle that we face with this. So how can we address these knowledge gaps? Looking at the data gathering, again, it always kind of goes back to the basic and preclinical science model. We just have to develop a better animal model, include consideration for sex as a biological variable. In diagnosis, we have to ensure that we are looking at cost-effective and sensitive diagnostic techniques, because if it's not sensitive enough to detect the problem for women and reaching a benchmark based on what is detectable in men, then there's going to be this whole unresolved issue of this disorder. And then at the clinical level, we need to ensure that there's a guide to managing ischemia without non-obstructive coronary artery disease, so that if the signs and symptoms that we've been accustomed to identifying differ, we're not just going to exclude those patients or individuals. And then the final example I'm going to go through today with respect to cardiovascular disease is SCAD, or spontaneous coronary artery dissection. And essentially what this is, it's a separation of the coronary arterial wall that can result in acute coronary syndrome, or ACS. Can also lead to myocardial infarction and potentially sudden death. And essentially what happens is that this blood vessel wall, a false lumen is generated. You can kind of see here, and blood is able to flow through that. And this can actually lead to spontaneous hemorrhaging. It can cause here this intramural hematoma, which can enlarge and encroach on this lumen space. And it can cause myocardial ischemia and infarction. So SCAD is commonly misdiagnosed or underdiagnosed, especially in women, and is typically managed as atherosclerosis. And it can actually, has been determined to potentially cause up to 35% of heart attacks or myocardial infarctions in women under the age of 55. And it has been linked to other disorders that disproportionately affect women. So for example, fibromuscular dysplasia, which is like the progressive twisting of different blood vessels, which disproportionately affects women, has been highly associated with SCAD. And pregnancy is also a huge risk factor, and they're saying it's suggested due to the amount of hormonal or the hormonal profile associated with this disorder. But the problem is, despite knowing that there are all these additional risk factors that contribute to this disease in women and how it disproportionately affects them, we still have not studied this well enough to understand how we can better diagnose and develop treatments. In fact, the only treatments essentially right now that are available, I think it's called like conservative treatment or whatnot, in which they just kind of like let the person to adjust lifestyle choices in hopes that it'll clear up in its own. But in terms of like proper therapeutics and drugs, there haven't been a lot developed to this point. So yeah, they typically will not provide a stent or a bypass, unless it's like super dire, but even then the definitions around what that means is kind of fuzzy. So yeah, there's just severe lack of diagnostics and treatment for cardiovascular diseases as well as other diseases that disproportionately affect women. So how can we address this with respect to SCAD? Again, data gathering. Biases in studies lead to misclassified SCAD episodes. We don't have a lot of predictors that prevent recurrent SCAD episodes that have been identified, so we have to improve that. You know, better reporting, better data gathering can improve and progress the science around it. Diagnosis, we have yet to develop a proper diagnostic tools, and we don't have enough studies being conducted on SCAD in young people without atherosclerotic occlusion. So we have to ensure that we're not just looking for the phenotypes we're used to seeing, but those that can potentially be indicative of progression of disease. And we have to focus on developing better therapeutics and defining long-term care for SCAD and not just conservative therapy. So kind of the major takeaways that I wanted y'all to get out of this was that sex differences in cardiovascular diseases are understudied, but they are increasingly important. The differences in onset of disease progression and clinical and subclinical diseases between men and women do exist, and they're important to recognize if we want to progress and ensure equitable and appropriate care for everybody. That there is a lack of appropriate diagnostics for various diseases that disproportionately affect women, and it's highly understudied, so we really have to make progress with respect to that. There is a need to include social and psychosocial determinants of health when studying sex and gender differences in CVD, because again, we're starting to realize it's not just a matter of genetics, but there's also an environmental epigenetic factor or component to health and disease that we have to account for. So despite evidence that sex is one of the most important modulators of disease risk and response to treatment, consideration for sex does not always translate to the clinic. Many medications are metabolized differently in women than men due to body size, distribution, sex hormone levels, enzyme activity. So for example, a couple examples of drugs would be the propranolol, I don't know if I said that correctly, but it's a beta blocker, and the levels are increased in women by about 80%. And there's this other sedative drug called zolpidem, if I'm pronouncing that correctly, and it's metabolized a lot more slowly in women. And even just fundamentally understanding these differences in the way that these drugs interact with the body between the sexes has a huge impact on how we should appropriately change the dosage between the two, and then what this means for patient outcomes. Women just generally have worse outcomes as compared to men across various CVDs. And the symptoms tend to be more subtle and often complex, and consequently, women tend to go without proper diagnosis. So we have to essentially understand the differences in the electrocardiophysiology and the therapeutics that are being developed. And right now we're at a point in time where we are starting to rapidly increase our understanding of, the genome and molecular basis of disease. And therefore, as we develop medications and therapeutics based on like gene therapy or how that can cater to, sex bias or sex-dependent differences, it shouldn't just be its own individual study, but rather this holistic sex is a biological variable, and its approach should be integrated across, any area of biomedicine and science. So just kind of to conclude off with the last slide, what exactly can we do about this? Step one, at the scientific discovery level, we have to ask how we can ground women's health in basic science. Bench research, which includes things like work with stem cells, cell lines, experimental studies, we have to explore sex at the molecular and cellular levels, and disease pathophysiology and prevalence extend beyond the hormonal influences, which could allude to the fact that each individual cell has a sex. Two, at the clinical trials level, we must ask if our discoveries are safe and effective for women. The testing phase has to include adequate numbers of women. It cannot just be centered around testing the physiology and the effects in men. And we have to ensure that we are not only conducting these experiments, including women, but also analyzing these trials and the reports based on sex. Step three is the translation of research into practice. We have to ask how we can use our research safely and effectively. At this stage, we have to use a sex and gender-specific lens in clinical practice. Healthcare professionals cannot be slow to adopt evidence-based guidelines. And then we have to address sex and gender in treatment, prevention, and diagnosis. And then finally, step four, measuring effectiveness. We have to ask what is the most effective way to diagnose and or treat women, and what is the value of this investment in women's health and just overall patient outcomes. We need to understand how sex and gender affect health outcomes, and if we want to achieve equity in research and practice, we have to ensure that we are measuring and reporting, measuring the reports of how we've integrated sex as a biological variable. So with that, I'm just going to conclude off by providing, a slide here with resources. This is the Silvera lab. This is one of the labs at UNC, and she was actually the one who provided a lecture kind of on the same topic about the importance of sex as a biological variable in one of my lectures. And I just think it's a great resource for us, or for anyone who is in biomedicine, science, who wants to be a practicing clinician. There's a lot of resources that she's compiled about how to better inform yourself and apply this kind of fieldless understanding to your work every day. And with that, thank you for tuning in. That was a little long, but thank you.